Bismillah Rahman Rahim, June 24, question paper 1-3, question 25 to question 40. Question number 25. Which properties of water are dependent on hydrogen bonding between water molecules? So cohesion had to be there. And then high latent heat of vaporization is because in order to break the bonds, it needs heat. Solvent action is because of the polar molecules making hydrogen bonds with the hydrogen and the oxygen. So that is not going to be there. So 25, that's why is B. Because solvent action is not dependent on the hydrogen bonding between water molecules. Question number 26. Which substances in xylem tissue are impermeable to water and prevent the collapse of the vessels? Impermeable to water. So the answer here is D. Why? Because impermeable to water is lignin, and prevent the collapse means what makes the wall of the xylem vessels, which is so sturdy and so strong that it prevents its collapse, would be cellulose and lignin, both in the wall. Cellulose and lignin, not lignin only. The cellulose, of course, also has a lot of immense strength because of all the bonding, the hydrogen bonds between the cellulose molecules. Question number 27. The electron micrograph shows a longitudinal section through the phloem tissue. Which student's drawing of a sieve tube element is correctly drawn and labeled? Now first let's look at all of them correctly. Now the one with the mitochondria is wrong. There could be no mitochondria inside. In fact, it's just peripheral cytoplasm. There's no mitochondria in the middle of it. So let's look at mitochondria. If there's any other mitochondria, this is wrong as well. So now we're left with choices of A and C. Now in this they've shown the cytoplasm as well because there is peripheral cytoplasm. So that this is correct. C is correct. In this one they did not show any cytoplasm. And the cell wall C plate is fine, but they did not show any details of it, which is of course the peripheral cytoplasm. Question number 28. Carrier protein in the cell surface membranes of companion cells are involved in the transfer of assimilates to phloem sieve tubes. The diagram represents the use of two types of carrier proteins in this process. So we have cell surface membrane, then something X is going here to here. So there's a 2 here and 5 here. And then we have X coming back and bringing along with it Y. So they've given you the key, there's a pump, and this is the co-transmotor. What are the substances labeled X and Y? So X has to be H positive ions, and along with it is sucrose, so it has to be A. Can't be glucose. Sucrose is co-transported with the hydrogen ions. Please remember that. That is something which you must know. It's not something that you can forget and sometimes say it's glucose, sometimes say it's sucrose. The diagram shows pressure changes during 2 cardiac cycle. Which row indicates atrial systole? So you've got to understand it's these speed breakers here. These are the atrial systole. Now that's not labeled here. It's labeled here. So what we have to understand is that the answer is B. You either know this or you don't know this graph. This has to be really clearly understood. Please take an effort to understand this. Find on YouTube a video which explains this very well, and then you can understand this better. I always remember it from the aorta. You see, this is the aortic this is the aortic pressure. So this is where the semilunar valves open at this point. Question number 30. An irregular heartbeat may be the result of ineffective electrical stimulation of the atria. Which area of the heart could be damaged, causing the irregular heartbeat? So which area of the heart? Now, you know, it says could be causing this irregular heartbeat. So atria, how does the whole thing start? Sinoatrial node is the pacemaker, initiates impulses, and then it spreads in the atria and then go to the AV node. And then from the AV node, it goes down into the bundle of His and then the Purkinje tissue. So which area of the heart could be damaged? So the answer to that is D, sinoatrial node, because sinoatrial node initiates impulses and they spread in the atria. So atria is talking about the atria. Question number 31. The diagram shows the effect of 3 different concentrations of carbon dioxide on the oxygen dissociation curve for human hemoglobin. So this is X is 3, Y is 5, and Z is 7. So the partial pressure of carbon dioxide is increasing. Now, what you have to understand is, I always say, is a very simple way of doing it. You have it here. Say this is 4 kilopascals. So let's draw a line straight from here. Now Z is and Y and X. So percentage saturation of hemoglobin, you can see what is happening to it. So percentage saturation of Z was say 40%. So we have this one would be 40% Z. Then Y is about say 50%. And then X is 70%. So you can see how it is percentage saturation is decreasing. That means from X, when it was 3 kilopascals, the carbon dioxide, from 70%, it has gone, when the partial pressure was 7 kPa, it's gone down to 40%. So that means the oxygen hemoglobin affinity has decreased. They were friends, now they're no longer friends. Now hemoglobin has given up its oxygen. It is less saturated. Please explain this to yourself in some sort of a way, like affinity. So oxygen and hemoglobin are good friends. But when carbon dioxide increases, they become less friends. So it's given up the oxygen. It is less saturated, percentage saturation of hemoglobin with oxygen. Now, if you've understood this, then only you can do this question. That is why we have to understand that the answer is B. It makes hemoglobin less efficient at taking up oxygen and more efficient at releasing oxygen because the percentage saturation has decreased. So the hemoglobin oxygen friendship is gone now. Only 40% of the hemoglobin is friends with oxygen. Previously, it was 70% were friends with oxygen. So please try to understand this. I know it's a difficult concept which needs to be clarified. Then coming to question number 32, which reactions will be taking place in blood in a capillary that is next to an alveolus? Well, it has to be only this one. Only one. That is why B is the answer. Now let's look at the other ones which are wrong. You see, this is going to happen in the tissues. The carbon dioxide diffuses and combines with water in the presence of carbonic anhydrase and form carbonic acid. The carbonic acid dissociates into bicarbonates and hydrogen ions. Hydrogen ions, of course, bind with hemoglobin to form hemoglobinic acid, which gives up its oxygen. Now that's happening in the tissues. So you must know what is happening in the tissues and what is happening in the alveolus. Question number 33. Which structure of the gas exchange system always contains cartilage? Trachea always contains cartilage, but there's no trachea here. So the answer is D. The bronchioles will not have, some bronchioles will have, but not all bronchioles will have cartilage. Then exchange of carbon dioxide and oxygen between air in the alveoli and blood in the capillaries of the lungs. Which partial pressures of the gases will allow gaseous exchange to occur? Now, question number 34. Exchange of carbon dioxide and oxygen occurs between air in the alveoli and blood in the capillaries of the lung. Which partial pressure of the gases will allow? So, of course, you've got to understand that carbon dioxide in the blood should always be more. So, these are wrong. Carbon dioxide in the blood is 6 and in the alveolus is 5.3. Only then diffusion is going to take place. So it was one of these, and here what has to happen? Alveolar air has to have more. Now this has to be more, this has to be more than this. Now this is wrong. This is 13.3 and this is 13.9, so it cannot happen here. Now this is 13.9 is a 5.3 oxygen in the blood. So then diffusion can take place from the alveolar air into this. So that is why the answer is B. You should first always see the ones which are wrong and then cross those out and then narrow down to the two choices. Then it says the plan drawing shows the cross section of a trachea. Which labeled tissue prevents the trachea from collapsing? Now the plan drawing, question number 35, the plan drawing shows the cross section of the trachea which prevents it from collapsing. Now it had to be A. Why it had to be A? Because you've got to understand is that they're showing you, this is the cartilage that's showing you. It's incomplete here. There's a muscle here. It couldn't be C that's complete. It's a C-shaped cartilage in the trachea. And D is the ciliated epithelium in the trachea. Question number 36. Which layers of cell does an oxygen molecule diffuse through when moving from an alveolus into an alveolar capillary? So alveolus has to have squamous epithelium, and the alveolar capillary has to be endothelium. So that's why the answer is C. Alveoli squamous epithelium, flattened cells, which are alveoli is made up of those flattened cells joined together like a jigsaw puzzle. And the alveolar capillaries are always made up of endothelium. So 36, the answer is C. Question number 37. Bacteria may be classified according to differences in cell wall structure. The differences are shown by using the Gram stain. The diagram shows a gram-positive bacterium and a gram-negative bacterium drawn to the same scale. So here you can see the gram-positive ones. They have peptidoglycan layer, then they have a periplasm, and then they have a cell surface membrane. The gram-negative is slightly different. They have the cell surface membrane, they have the periplasm, but then they have a sort of a thinner peptidoglycan layer, and then they have another periplasm, and then they have an outer membrane made up of carbohydrates and lipids. These are very difficult to kill, these gram-negative bacteria, because of these entire the layers around it, which is of course a big protection to them. Now I would say the antibiotic penicillin kills bacteria by inhibiting the synthesis of cell walls during bacterial cell growth. Which type of bacteria will be killed by penicillin more easily and why? Now basically it was very simple. You see, because the cell wall needs to be destroyed. Now here the cell wall, the peptidoglycan layer is then has another layer, another periplasm and outer membrane made of. So it's really not going to be easy to kill this to reach the cell wall. So that is why the answer is A. Gram-positive bacteria, because the peptidoglycan layer is exposed to penicillin directly. How will penicillin reach in this one, how will it reach this layer? It has to first pass through all these things and then reach this cell wall. So please understand why this was difficult. This was a little difficult question. I mean, I would find this a very, very interesting to see the report on it. How many students got this correct? Question number 38. Which facts relate to the disease TB or its pathogen? Viruses. No, sorry, TB is B for bacteria. It's not a virus, so I would cancel this out. TB is caused by only one species of pathogen nodes called the Mycobacterium bovis and Mycobacterium tuberculosis. HIV/AIDS makes the bacterial infection worse. HIV lowers your immune system. The pathogens may be transmitted by ingestion. The pathogen may be transmitted from animals. Multi-drug resistance occurs. So now which facts refer to the disease TB or its pathogen? Now the answer is D, but why is the answer D? Let's look at it. Yes, immune system weakens, so 3 is correct. Pathogen may be transmitted by ingestion if you have raw, uncooked meat because Mycobacterium bovis is found in cattle, and so that can be transmitted. The pathogen may be transmitted from animals. Yes, animal meat. I'm not saying from animals directly, but animal meat and multi-drug resistance forms do occur in it. Now, 1 and 2 are the wrong ones. Virus, of course, is the wrong one. The first one, so anything with 1 was wrong, so these 2 were wrong. Now we narrow down to 3. Now, why is 2 wrong? Because TB is caused by only one species of pathogen. This was wrong. TB is caused by Mycobacterium tuberculosis and Mycobacterium bovis. So then if you ruled out 1 and 2, you knew wherever there was 1 or 2 that was wrong, so you're only left with D. That was a very easy way of doing it. Look at the ones which are totally wrong. 1. So 1, these ones, so this was wrong. Then 2 is wrong as well. So where 2 is present, then that's wrong. So you see the one without 1 and 2. Question number 39. The events listed occur during the primary immune response to a specific pathogen. Activation of B lymphocyte to produce plasma cells and memory cells. Phagocytosis of invading pathogens by macrophages, T helper cell activation and production of T killer cells, expression of antigen on phagocytic cell surface, production and release of antibodies. Now, I would have sort of thought of it when I was doing this question for the first time. I thought 5 should come at the end. And so I narrowed it down to B and D. The answer is B. Why is the answer B? Because you see, look at it. 2 is what? Phagocytosis. So this is the first one. Phagocytosis invading pathogen by macrophages. Then what happens? 4. Expression of antigen on the phagocytic cell surface. It becomes an antigen presenting cell. Then 3. T helper cell activation and production of T killer cells. Then 1. Activation of B lymphocyte to produce plasma and memory cells. And then production and release of antibodies. So I knew this, that 5 should have come right at the end. Production and release of antibodies is the last thing in the immune response. Then coming to the last question, 40. Influenza is an infectious disease caused by a virus. Fine, they're telling you this in the question. Is it possible to have influenza more than once? Which statement explains why it is possible to have influenza more than once? The viral antigen change as a result of mutations. The immune system may be weak and make few B memory cells. Untreated HIV infection has resulted in a low T helper cell count. But what did you say in the question? Is it possible to have influenza more than once? Now you see, the question says influenza more than once. Why would I have the influenza more than? There can be 2 reasons. Number 1, my immune system is very weak. Number 2, the virus for influenza is of many strains. There are many strains of the virus. All of them cause influenza, but what is different on them? They have different antigens on them. But they all cause influenza. So the viral antigen change as a result of mutation. So 1 is correct. Then they write the immune system may be weak and make few B memory cells. So I had influenza, lasted a week, and then I'm better for a week, and then I get it again. Because I have a weak immune system. Untreated HIV infection have resulted in 2. Yes, that's right. If somebody has HIV, then they have a low T helper cell count, so their immune response is very, very pathetic. So they will not be able to respond to the infection, and that can really then result in some other problems like superimposed bacterial infections or secondary bacterial infections. So that is why the answer is A, which is 1, 2, and 3. Because they've said, is it possible to have influenza more than once? And which statement explains why it is possible to have influenza more than once? One, of course, is the fact that you have the virus is different. The strain of the virus is different. You went for Umrah or Hajj, and there people are coming from all the different parts of the world, and everyone is bringing a different strain of the influenza virus. So many aeroplanes are coming. They're bringing in a whole lot of different strains of the virus from all the different parts of the world, from America, from Australia, from UK, from Dubai. So you've got to understand how this question would be handled.